Welcome to The Explainer. Today, we're diving into something that sounds like it's straight out of science fiction, the science of genetic engineering. We're going to look at how scientists can literally rewrite the fundamental code of life. So, let's start with the big problem, right? Think about it. A living cell is basically this tiny, microscopic fortress. It's got all these defenses designed to keep intruders out. So if you're a scientist who wants to get in and change the instructions, well, first you have to figure out how to get past the gatekeeper.
And just how tough are those defenses? Let's take a closer look at what we mean by this cellular fortress.
Okay, so there's a technical name for this whole operation. It's called transfection. All it really means is the process of getting new genetic material, like DNA or RNA, inside a cell. And why would you want to do that? Well, it lets scientists watch a gene or a protein in action, right in its home environment, to figure out exactly what it does. Now, once you get that new code inside, one of two things can happen. First, you could have what's called a transient transfection. Think of this like a short-term visitor. The new DNA comes in, does its thing for a little while, and then it's gone. But then there's stable transfection, and this is the big one. The new DNA actually integrates itself right into the cell's own genome. It becomes a permanent part of the blueprint, getting copied every single time that cell divides.
So, the big question is, how? How do scientists actually pull this off? Well, they've developed this absolutely incredible toolkit to get the job done.
Broadly speaking, there are two main approaches. You can use viruses. They're nature's original experts at smuggling genetic code into cells, right? Or you can go the non-viral route, which uses either chemistry or just raw physical force. And today, we're going to zero in on a couple of those amazing physical tools.
All right, tool number one. This one is all about precision. I want you to imagine performing surgery on something you can't even see with your own eyes. That's basically what we're talking about with a technique called microinjection. The idea here is, well, it's pretty straightforward. You literally take a tiny, super fine glass needle, fill it with the DNA you want to use, and then you physically inject it right into the heart of the cell, the nucleus. It doesn't get much more direct than that. And here's what that looks like in real life. It's wild, isn't it? On the left, you've got this little pipette holding the cell in place with a bit of suction. And then from the right, here comes that microscopic needle ready to pierce the cell membrane and deliver the goods. It is, quite literally, surgery on a single cell.
The whole process is this incredibly delicate dance. Step one, you've got to hold that cell perfectly still. Then you bring in the needle, which can be as thin as half a micrometer, just think about that, and you pierce the cell. You inject the DNA right into the nucleus, the control center, and then you have to pull the needle out without wrecking everything. You can imagine every single step takes a ton of skill and some seriously high-tech gear. So you can probably see the trade-offs here. The huge pro is that this method is super precise, and it's great for getting that new DNA to stick around permanently. But the cons? Well, it's really expensive. It's incredibly slow. You're doing this one cell at a time, and you have to be a true expert to pull it off.
Okay, now let's do a complete 180. If microinjection is like a surgeon's scalpel, our next tool, well, it's more like a cannon. And I'm not kidding, that's not just a metaphor. The technique is technically called biolistics, but everybody just calls it the gene gun. And yeah, it does exactly what you think it does. It literally shoots genetic material into cells.
So how does a gene gun actually work? First, you take these tiny little particles of gold or tungsten, basically microscopic bullets, and you coat them with your DNA. You load these DNA-coated beads into the gun. Then, a powerful blast of compressed helium gas fires these bullets at an insane speed, fast enough to punch right through the tough walls and membranes of the target cells. The big plus here is that it's fast and relatively simple. You can hit a ton of cells all at once, and it's especially useful for things like plants that have really tough cell walls. But the downside is pretty obvious, right? It's a brute force approach. You're blasting cells. So yeah, it can cause a lot of damage, and you can only really get to the cells on the surface.
So, we've got these scalpels and we've got these cannons. Amazing tools for rewriting genetic code. But what's the point? Why does any of this actually matter? What can we do with it?
And the scalpel and the cannon are just the beginning. The full toolkit is huge. Scientists can use viruses, as we mentioned. They can use special chemicals to trick the cell membrane into becoming a little leaky. They can even use something called electroporation, which basically zaps the cell with electricity to open up temporary little holes.
And this power to edit cells, I mean, it's changing our world. This is the technology behind genetically modified crops that can fight off diseases. It's the basis for gene therapies that might one day cure diseases we're born with. And for researchers, it's the key to unlocking the deepest secrets of how life even works. Really, what it all comes down to is cause and effect. If you can add a new gene or tweak an existing one, you can sit back and watch what happens. It's how scientists are piecing together the giant puzzle of our DNA, figuring out what each and every gene is for, one piece at a time. So we've reached this incredible moment in history where we can literally edit the book of life. And the tools are getting better every single day. We're moving past the cannons and scalpels to even more precise tools like CRISPR. The science is just breathtaking. But it also leaves us with a massive question. Now that we have the power to write this new story, what story should we write?